morning, everyone. My name is Sidney Sudberg, and I am the founder and CSO of Alkemist Labs. I'm here to talk to you today about a very interesting subject, and it's called ayahuasca. But first, I wanted to thank the organizers for inviting me. I am deeply honored to be able to present to the HPTLC Association and its members the interesting and exciting research we've done over the past several years on this interesting and novel medicinal plants, medicinal plant combination. Um, we are using HPTLC primarily, in addition to MS, HPLC, microscopy, macroscopy, to create some type of analytical predictive method for the improvement of and the identification, characterization, but more importantly, the therapeutic efficacy. So let's get into it. What I am trying to get across here today, I want to let you know that initially I will just give you some information about this material called ayahuasca, and from there we'll go into some identification characterization slides, and after that I will give you some conclusions or findings that we have found to be important with respect to the ability of HPTLC to assist in the process of improving um, therapeutic efficacy. So what is ayahuasca? Ayahuasca is a psychotropic Amazonian beverage formulated from the water decoction of Banisteriopsis caapi vines and Psychotria viridis leaves. This drink, this tea, this brew is legally used in Brazil and Peru, as well as some of the European countries and the USA. Now, primarily it's used traditionally for religious, cultural, and therapeutic purposes, but its effects can be described as displaying potential efficacy within a model that involves currently biomedical, psychological, and anthropological, as well as theological elements. It's believed that the psychoactive effects of ayahuasca can exhibit the potential for the treatment of mental disorders due to the effects of the beta carboline derived alkaloids present in the vines and N,N-dimethyltryptamine, DMT, present in the Psychotria leaves. Because of its effects on the nervous system, as well as the presence of alkaloids, ayahuasca is classified as psychoactive and has been also described for many years in the West, or not in the West, but maybe in the North, because most of what has been described so far has been in South America. But it's been described as psychedelic or hallucinogenic. I know all of you have heard these terms. It's become quite popular recently, and for a good reason, more than likely. And we're finding that there is a lot of interest, and because there's a growing number of studies indicating the potential increase in ayahuasca in general addiction treatments, namely alcohol, tobacco, illicit drugs such as cocaine and heroin, as well as in the treatment of depression and anxiety. Now, to get into more detail of the preparation of ayahuasca, typically the washed leaves of Psychotria viridis and the smashed stems of Banisteriopsis caapi are placed with water in a large kettle, typically 10, 20, well, typically 20 gallons or more, and decocted for about an hour or two initially. And this varies from individual to individual, whoever the ayahuasquero or shaman is that's preparing it in South America. And then after that, new batches of both plants are placed again in the kettle, and the first decoction, or the liquid from the first decoction, is added to that. From this point on, there's a considerable variation in boiling time, concentration of the brew with evaporation, and then the reinforcements with the new plant material. Now, this is an important factor that I'm about to tell you. The subjective effects of the ayahuasca are tested by volunteers. Now, they're selected by the person who's responsible for the preparation, and this is to assure and in order to attain an expected range of the ayahuasca experience. You could say not very scientific, but in a sense it is. It's collection of data, it's qualitative, and it's real. And it is um, predictable, but not necessarily well controlled. So it's important to note that there are different elements uh, uh, that add to the ayahuasca variability, and that would have to be considered, as in alkaloid concentrations in the plant material, the starting materials, amount of water, decoction time, and the number of reinforcements of the decoction with the new plant material. One final thing before I get into some data and data analysis, the term ayahuasca comes from the Quechua culture of South America and can be translated into English as vine of the souls. So that gives you a sense of what this material has to offer. So the goals of this study, we started by trying to replicate the actual manufacturing that takes place in the jungles. And uh, there, like I described earlier, there's what I am defining as the weight method. In other words, you take certain proportions of the leaf to the vine, you boil them, etc. And then you ultimately end up with what would be called, depending upon who's making it, either a tea, could turn into quote unquote a sludge, which is somewhat molasses-like, and that, of course, is going to have an effect on its dose of the various compounds that we'll be looking at momentarily. Then there's a volume method or volume method, which we try to replicate in the lab. Um, the volume method is where each of these individual plants is individually boiled, decocted, evaporated, and concentrated, etc., and then combined to produce the ayahuasca mixture. So our goal with this was to see if either one of those had an effect on the actual extraction and or the final um, analysis of the various compounds, the beta carbolines from the Banisteriopsis and the N,N-dimethyltryptamine from the Psychotria. And ideally, our goal, which we didn't realize at first, was to discover a ratio of the active compounds that could be optimized by this method, especially because TLC is such a perfect fit for this purpose, um, where we can look at the ayahuasca without getting into the the details, and so that we would not miss the forest for the trees. It has such an elegant way of producing information data that can be utilized in a predictive way. And that's what I'm trying to demonstrate. So first of all, we're going to be looking at the different individual compounds. Well, before that, we'll be looking at the fingerprints, the identification, and then the characterization of the chemical compounds, and then comparison of the two methods of preparation. And finally, we'll look at some other data that we collected several years ago. So what is the ayahuasca comprised of? Banisteriopsis caapi, a vine, as you can see here on the left with the upper left corner, the vine looks similar to that. It could be varying in anywhere from 1 to 6 inches in diameter, each of those those actual stems. And the compounds that are derived from it are harmine, harmaline, and tetrahydroharmine. There's also harmol and harmalol, but they're in very low concentration and not thought to be as active as these three compounds. In particular, harmine, where that is the kind of the parent compound of these. And within the preparation of the ayahuasca mixture, the data has suggested that prolonged heating and boiling of the ayahuasca preparations leads to this interconversion of some of these components, in particular these three components, starting with the harmine. It appears that to occur to a significant degree for the changes seen that result in tetrahydroharmine from harmine to harmaline levels between samples that are undergoing further heating necessary to attain higher concentrations of the components. But then there's a backlash and an effect on this the concentration of the active component, being primarily harmine. The phenomenon, an apparent acid driven chemical reduction of harmine to harmaline to tetrahydroharmine has also been reported by other investigators, Callaway in particular, references at the end of my presentation, and it constitutes another source of potential variability of results among ayahuasca preparations and preparers. Depending upon who's preparing, they may decide to make it more concentrated, but at the same time, that then reduces the effects. So more on that later. So now the next plant is the Psychotria viridis. The leaves are there on the left, and the dimethyltryptamine is structure is here, an indole alkaloid, also the hallucinogenic component of this mixture. So we then went into fingerprinting of by HPTLC, where we have, in this particular case, 15 lanes, and in lanes 2 through 5, we have different various accessions of Banisteriopsis. And then in lanes 7 and 8, we have Psychotria viridis. And in 9 through 11, some other species of Psychotria, and then two other plants, Diplopteris cabrera and Mimosa hostilis, known to have content to contain DMT. And you can see in lane 14 at approximately the green band, approximately 0.45, that there is DMT, and it looks like it may even have increased concentration. So it may even be another source for this plant's use, for this mixture's use. So here we have our botanical test method that we create from this uh, previous slides, where we actually characterize and are able to distinguish with some degree of specificity among the different species, and or reflect some of the quality that we're looking for in terms of just looking at a starting material and seeing if there's anything worth looking at, and to then take it further and then do quantitative analysis. But before we do that, we would look at the microscopy. We would also characterize the stem of the Banisteriopsis. We would look at it microscopically, as well as looking at the HPLC content, or quantitative content of those various compounds. Same thing goes for the Psychotria, where we have the macroscopy, the microscopy, and then with the HPLC analysis, looking at the DMT content. So we've done this, many times and over the years, and there are some studies that we're participating in that we need to get more data for, and we wanted to have a fully characterized, at least as fully as possible, characterization of these plants, so that when this goes to clinical trials, all the dots are dotted, all the I's are dotted, all the T's are crossed, etc. So in this slide and these subsequent images, I'm going to show you where those various beta carbolines appear on the chromatogram, and where you see at the point 1, the harmol and harmalol do coalute. And since they're not necessarily important at this point, we don't really have much concern for them. Most importantly are the harmine, which is showing here at about 0.4, and then the DMT, which is showing at about almost 0.5. So from here, we went into the characterization of the individual reference standards using, um, obviously, HPTLC. And with this particular um, experiment, we uh, provided a calibration curve. But first, we characterized the harmine reference standards just to get their UV maxima by scanning with the scanning densitometer. And we had the video densitometry to give us a relative degree of, or relative quantitation, qualitatively speaking. So we would be able to compare from plant to plant, and as well as the MS um, spectra that we did with our Advion compact mass spec. We were not able to scrape the plates in this particular case. Our pump was down, and we're still working on that. But at this point, these were taken directly with the ASAP probe, and directly solutions were directly not ejected, but got into the mass spec, and then gave us the spectra that we were looking for. Same thing with the DMT. We see here, um, that we have the DMT characterized with a maximum of 223. Again, scanning, densitometry, video densitometry, mass spec. Then we looked at the individual plants and we did the same basic procedure, scanned, did mass spec, identified compounds, characterized them, as well as we quantitated from these with the actual from the actual Banisteriopsis, Psychotria, and then onward to the ayahuasca, where we did our final kind of experiment looking at the ayahuasca with the weight method, the weight, lanes 511, water extracts. So we did some, again, we scanned with scanning densitometry, and we then did the same thing for the volume method. And we get numbers. We get a calibration curve, a good correlation coefficient, etc. Uh, Low coefficient of variation. And the numbers I'll kind of talk about in a moment, but the numbers will be reflective that the harmine water extracts um, uh, have a specific relationship with the volume to the um, weight method. So just added one more experiment in there because I wasn't too happy with the content that we were getting, and it really did reflect that we might have been shy on the content with respect to the water extracts, the way we were preparing it. So we did methanol extracts with a more exhaustive extraction and found that we actually did did attain or obtain much higher concentrations using methanol. But that aside, that just gave us an idea of what we should have expected, and we know we have more work to do on the method that we would use if we were to try to reproduce this in the lab. But basically, if you look at the top table there with the percent DMT, both with water and the weight to volume comparisons you see in the 0.3261, 0.3105 versus 0.239 and 0.9, 0.2611. So basically, the water extraction for the DMT is higher than the volume method of extraction. You go over to the right where it's a percent harmine after the methanol, although the methanol is interesting because you can see the methanol extractions were almost double what we did with the water. And in the harmine concentration, where the weight to the volume methods were compared, we have 0.06 to 0.02 so percent. And this is demonstrating that the the volume extraction was greater than the 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 weight method, the combined method. And again, the methanol extractions show a much higher content of the DMT, I mean of the harmine, with respect to the water. This slide is really the pivotal slide for me and what led us to be able to develop this research so that we can actually do what we are intending to do. So if you go to the bottom of the left side, the table, and you go up a little bit, the fifth and sixth, and the seventh and eighth up, and it has 5 stars, 1 star, 1 star. So if you go to the right where the 1 star is, you see that the content, the percent DMT of the 1 star is 0.69 and 0.65 versus the percentage of harmine 0.89, 0.77. And if you go to the 5 star, you see that the 0.58, 0.56 has a 1 to 2, or if you go harmine to DMT, a 2 to 1 relationship, as opposed to an almost 1 to 1 relationship. Therefore, this uh, this reflects what I've been looking for, and we are going to use the HPTLC to help us achieve this uh, ultimate goal. I did not explain yet what 1 star and 5 star mean, and I apologize for that. But the 1 star is the low, very poor effects, almost like a dud. And the 5 star are extremely powerful, extremely beneficial, extremely therapeutic. So hence, we found that the ratio of harmine to DMT suggests in the 5 star and the 1 star, and it goes throughout the rest of that table up there. should be 2 to 3 times the concentration of DMT. Um, and what you're looking at here is that the 2 to 3 times the concentration of harmine to DMT, while the DMT should not be less than 0.5% DMT. And this is something that we can easily do and achieve by using HPTLC, even just visually at some point. But we do have the scanning densitometry and the ability to not necessarily miss the forest for the trees, because everyone that's doing research right now is collecting information mostly on the exact content of these compounds, which is academically interesting and important, but therapeutically and practically speaking, uh, I think we are more than it's more than adequate to do what my premise is, and that is to obviously ultimately create optimum concentrations and to figure out how to create the optimum concentrations for the highest quality experience when it comes to the beverage, the hallucinogenic beverage ayahuasca. I thank you for your attention. It has been my pleasure to give this talk. I hope that I can get some interesting and questions and possibly even some comments and suggestions. So thank you again. Very special thanks to Con Tran, Nam Huang, Tori French, and Anthony Fontana, because without their help, this project would not exist today. Thank you.